This is the very last lecture of Chem 225. Thank you for listening all this semester, and congratulations in reaching to the end. So I'll try to make this as brief as possible, but I just wanted to finish up. We talked about the sickle cell anemia, and I wanted to make sure that you are able to answer all these questions that are listed on the drill, and you should be able to answer these questions. But the part that I have not gone over is the hydrolysis of peptides, and also when a peptide is denatured, all right? And you need to know the difference between the two as well. So when a protein is heated with acid, the peptide bonds are hydrolyzed and amino acids are produced. So this is reverse of peptide synthesis, obviously, and this is what happens when you eat a protein food. All right, so let's just write protein hydrolysis, and this is when, let's say we have tripeptide such as this, and you treat it with acid and water, then or you could have it in under basic conditions as well, as you know. Then you get alanine, glycine, cysteine. These are free amino acids, and so you're breaking up all the amide bonds. All right, so the protein digestion is enzyme catalyzed hydrolysis in our body, and the free amino acids, when it's released, is absorbed through intestinal wall into the bloodstream and transported to become raw materials for synthesis of new protein, or it could be metabolized totally. All right, now protein denaturization is something totally different. What this is a disruption of secondary, tertiary, and quaternary structure of a protein. All right. So pictorially, this is what it would look like if you denature a protein. Is you're not breaking up the amide bonds, but you're getting rid of any of the hydrogen bonding, anything that's responsible for the 3D structure of the protein. So denaturization of, for example, alpha helix would unravel that orderly alpha helix. And it would give you random structure, all right? So this is very, very important, because disrupting the secondary, tertiary, quaternary structure, this causes loss of 3D shape of protein, which in turn causes loss of activity for that protein. So this is something you want to avoid in your body, because we want the enzymes to work the way it's supposed to, and we don't want to denature them. So what causes proteins to denature, right? I'm going to write down at the very bottom. I'm actually answering this question: what factors force denaturization in proteins? Okay. First factor is heat. So this is what happens when you're cooking meat, eggs, whatever, right? When you're giving a lot of temperature, heat, then it is causing the denaturization to occur. So same thing could happen to you if your body temperature rises above 106. So that's why when you have a fever, you take a temperature, make sure that it's not really, really high, because if it if you're really high, you have a high fever like this, then you could start to denature your enzymes, which would be very dangerous. UV radiation is also bad for you. This is how you could denature proteins in your skin. So this is why, one of the reasons why it's not recommended that you go and burn yourself under the sun. Now that you know free radical chemistry and know that denaturization occurs, this are, you know, two reasons why you should try to avoid UV rays as much as possible. All right, and the third is acids and bases. So this should make sense. I mean, you know, these are the conditions that we use in lab to break up the amide bonds. So initially it's going to also break up the shape as well. So this is why it's very important that you wear safety goggles in lab, because you don't want the acid or base to harm your proteins and what's important in your eyes. And then some metals cause denaturization, especially mercury, as you know, and lead. You've heard these before, and so you want to avoid poisoning of these metals. What happens is that a lot of these metals are attracted to functional groups. So because the 2D and the 3D-ness of protein is based on a lot of these electrostatic interactions and hydrogen bonding, it could disrupt that and causing the shape to unravel. And the last is alcohol. So for example, isopropanol, ethanol could disrupt it as well. And this is why when you get an injection in the doctor's office, they usually swab your skin with alcohol before giving injections. So because the alcohol denatures the bacterial surface, and so it kills them, and therefore you won't have infection as you puncture your skin. So that concludes the protein lecture, and you should now be able to answer all the questions that I've assigned you. That's it. I am this is a new method of teaching, so I hope that these lectures that are videotaped are helpful. I will be asking you to tell me your opinion as to, you know, this method versus the normal method, what you think is more effective in the last lecture. So keep that in mind before you come to the last lecture as well. So once again, congratulations in reaching the last lecture of the semester, and good luck in your final at the end of the semester.